Today I want to talk about homology-based inference in context of the CAFA competition. And this simply is one example of how homology-based inference works. The slides today come from Tobias Hamm, who in fact studied computer science, went into computational biology, did his master in bioinformatics here in TUM, did a research stay at Columbia University, did a PhD here at TUM, some postdoc, and now is at the Alumni in Cambridge, England. And has quite some publication. He's an expert in machine learning. And we will encounter him later during the talk, during the lecture. So let's just begin and repeat here. So there are these typical things that make life easier, and molecular biology simply researches life on the level of the molecular level. And one thing is very, very clear, and I believe maybe I've given you the wrong idea. We do not understand how life works, right? We know how proteins fold, we know how many of these parts fit together, but we cannot compute a cell. We cannot compute life. And there is so much unknown that in fact I'd argue the amount of things we know is much lower than the amount of things we don't know. And that's what we sort of realize in this field every other year. Google has moved already quite a few years into this field because it is clear that the combination of data and biology, computers and biology, is something that clearly is important for the years to come. For health is one of those issues. In some sense, the problem here is that if you look at all of these machines, they have a common background, they have a common functioning. And the question actually is, we assume that they have a common functioning too. If we say on the base of life is DNA, there are proteins, there are cells, and all of that, that is the same. But then ultimately that doesn't define your computer, right? Your computer is defined by somehow how these parts are put together in a particular way, and how that is connected to the software. And what is the analogous situation on the level of life? We don't understand. On a simplistic level, where we have DNA, so this is what you typically see in images, this is actually the way it's sort of rolled up in the cell, it's densely packed. Cells have several compartments. They form organs. The scales are around about here. So biology happens on all of these scales. That's another important thing. So on the level of a single cell, that's bacteria. But a lot of things happen. So this is the part where we don't understand life. Okay? For us, these other levels are very important, and we clearly don't understand those. So the lack of knowledge is really very, very big. Yes? Yeah. So as long as I don't move, I believe it will work fine. So I have a very static talk from now on. The way, again, we cannot predict protein structures. The way we approach it is that we build essentially energy functions. I said that you take a protein sequence, you throw it into a solvent, and it adopts a unique three-dimensional structure. So there is somehow an interaction between the environment and the internal forces. And this is an attempt to model the internal forces. You essentially look at every single atom or every single molecule and ask, what is it influenced by? And build up a simple potential here that looks at harmonic bond, bending, the rotation of a bond, and then the interactions of a bond. They're modeled in a simple way. Okay, if you like that one. And then they form the protein. Should we give her that? Yes. So you already see that if we wanted to understand the entire machinery from the bottom up, we're very far away because we cannot even understand the protein's fold. A shortcut could be essentially that we gaze at the genes, that we see there's something different between the two, any pair in the room, and from that difference of us here in the room, we can infer something about phenotype. Why we look different. Why we have different hair, why we have different, all kinds of different macroscopic features. And in fact, that is one of the most important issues that the computational biology is currently addressing. Can I get from sequence variants to phenotype prediction? Where exactly do we stand? Well, in some sense, Tobias said it this way here, put down differently, the data is increasing a lot, and we don't quite know what to do with it. And that has to do with the limitation in the devices with which we can measure. We can, in fact, get our model organisms, the experimental speed. So we talk about what's the effect. Later we'll talk about the prediction of an effect of a variant, a sequence variant. Okay, we're going to talk about how to develop a method that predicts sequence variants. For every single one of those to do the experiment may take months or years. And that is an issue. There's a lot of experimental noise, so they're high throughput experiments, but they're getting noisy. Even low throughput experiments are very noisy and very costly. Again, so just a few years ago, to determine the impact upon molecular function of one particular variant did cost something in the ballpark of 3 to 5 years of a person, and you can sort of add up these costs. Plus, there's a lot of wet lab costs. Computing power is in fact something else that is highly in demand because the biological data is growing much faster than computers. More and more, biology is becoming a truly probabilistic model or probabilistic view. In the past, typically biology had designed an experiment, and for that experiment they measured that a protein binds. The readout is bind, yes, no. And that was relatively noise-free. That was relatively clear. Today, typically what they do is they look at 1,000 proteins, whether they somehow bind, and for every one of those they get a probabilistic statement. So we're getting to the point where, in fact, the line between experimental biology and computational biology is fading. It's getting narrower. The distinction is getting more difficult because any experimental data will be interpreted by computers, by models that are underneath, probabilistic models that are underneath. Okay, let's get back to the main issue here. Now, before we get into the main issue, again, what is a protein? A protein is a unit that comes from a gene. They're building blocks. Building blocks are, in fact, on the smallest level is written a residue, on an amino acid. On the higher level, you see secondary structure segments, here the helices. And on an even higher level, you see domain architectures. You could see these are two different domains. They come together. Again, the biological data grows much faster than the CPU speed. Many proteins are enzymes, and we talked last week about enzymatic classification through homology modeling. And the protein is, in fact, again, the sequence-determined structure. It is a solid three-dimensional structure, always the same, but it moves. It's dynamic. And the way it moves is very crucial for the function. It's not a rock. In some places it's more mobile. So an example is here, the region in red, upon binding, adopts a completely unique, different three-dimensional structure than in this point. In fact, this kind of thing is called disorder, because here, if you showed light at it at two different times, again, we cannot do that. There's a Gedanken experiment. So if you looked at it at two different time points, you would see two different things. You look at it at two different time points here, you see the same. So upon binding, it adopts a unique three-dimensional structure. And now we get into what came to this point a few years ago in a lecture. And in this lecture, we had 16 students, and we 16 students, we split into two groups. Now, I thought that I defined what I wanted to do. Ultimately, there's a slide missing here. Ultimately, the idea is I build a database of proteins of known function. Every protein here is a stack. Every protein here, I know something experimentally about its function. And one particular way of defining function, I will get back to that in a moment, is the gene ontology, GO, Go. So Go is a simple number, or in fact 3 numbers, that describes what we experimentally know about that protein. Okay, and in this database, in this set here, for every single one in there, I know the function. Okay, now I start with the protein U, for which I do not know the function. And I'm going to run an alignment and I'm going to essentially see for every single one, what's the score? How similar are they? So the simplest way of measuring sequence similarity is percentage sequence identity. So you count how many letters are identical. So the 20 amino acids, how many, when you superpose two sequences, how many are identical, okay? Another way would be an e-value. We talked about that, and then we also talked about the h-value. So the distance from the h, this is p-curve, where this is the length of the alignment here. This is percentage sequence identity, PIDE typically. And there was a curve that at 100 residues here is sort of 33. We now can look at the distance from this curve. This is plus and this is minus in this curve. Those are three different measures by which I can measure sequence identity. So for every single one of these hits here, in this particular case 5, I get a similarity measure, okay? And now, how do I predict function? Homology-based inference, what do? It's very simple, just say it. And if you feel it's not simple, then please tell me too. Am I overloading you, or is it too trivial? So every single one of these here has a number that I know. This is go 1, 2, 2, 3, 4, 5. Okay? Those are 5 different go numbers. So they look different, the real go numbers. I'm just too lazy to write them. Here's my protein for which I do not know the go function. I want to, yeah?
Pick the 5 most matching, and then infer by majority vote or something like that. Like the function that...
Yes. The problem is you already killed one of my... So I gave this lecture, and initially I really was dressed without a dog. So I simply said, find the one with the highest similarity, and that one you take. That was my definition. Okay? So you go a step further, and that going a step further is something that I'll get back to. So anyway, initially that's what we said. We we essentially split the group, the students in the exercises into 3 different groups, and but you stay here, right? No. Thank you. Thanks so much. Okay, so let's just look at what we do. Again, function is anything that happens to or through a protein, and we define, let's get to the next point here, we define function as Go category. Go has 3 different levels. There's the biological process, essentially what pathway are you part of? Then there is the cellular component. This essentially means localization. Where are you in the cell? Okay, somebody's proud. And then there's molecular function. The molecular function is, for instance, the enzymatic activity. It is an enzyme. What type of reaction does it catalyze, for instance? So these are 3 different numbers. 3 different Go numbers. So every entry here may have up to 3 numbers. Sometimes one of these numbers is not known. It may be known that's an ADH, but it may not be known in which pathway. Glycolysis pathway, for instance, it participates. Okay, so again, then we have a lot of proteins in our database. We have sequences, and we identify the similar sequences. Here's the slide that I was looking for, just at the wrong place. That's exactly what I write here. Okay, now the one way of doing it is in principle, you already alluded to that here. We could possibly, in fact, look at the not nearest one, but a few nearest neighbors. You said 5. That's where k nearest, where k is 5. Okay, here's the first group. And in fact, I said in my lecture, I defined what I said here, simply take the best one. That was the goal. And then the goal was to participate in CAFA. Now, CAFA, I will go over it a little bit later, is simply a competition that tries to measure how well do we do in terms of function prediction. And I felt the simplest thing we can do is something like this. Very well-defined problem. Let's just see how the sort of dumbest thing can contribute to that competition. We never tried to really compete big because this was so simple that we felt, anyway, we wanted to define that. Now here's the translation of my take the best of the first group. In fact, they took a weighted 6 nearest neighbor, so it's like your 5, but I put a weight in there. They considered hit count. So in fact, typically you can say there's a threshold. If the percentage sequence identity is so low, I'm not going to consider that. If it is high enough, I call it a hit. So number above a certain threshold in terms of sequence identity is a hit. Okay? And they felt that even if I only consider 6, whether I hit a function that has 50 members or 100 or 5, in total, maybe the 5 is the total number, and maybe the total number would be 50, and I take the best 5, and that should differ. That should make a difference, right? So that information should somehow come in. The method had 3 output, 3 terms per ontology, meaning that for every 3 of these Go categories, they had 3 different answers. So they took the top 6 and condensed them to 3 different answers with these output scores. In sort of more formal terms, essentially they predict 3 different trees from the 6 first hits that they have. The second group, again, did the same thing, did also a weighted k nearest neighbor, had a different scoring scheme. So the scoring scheme essentially is, how do you compile whether or not 2 proteins are similar? I said you could use an E value, you could use an H value, you could use a percentage sequence identity. Maybe you can use all of them. Maybe you somehow can combine that. And that was the idea here. Then the score was normalized in a clever way, and then they began to optimize a couple of parameters. So ideally, in principle here, this is the background score. The optimization was done over the tree. Again, Go is a tree. Okay, the third group did a weighted E-value based nearest neighbor, some custom scoring scheme on continuous term scores. So you have an eye on her on the cables? Just watch for the cables.
Oh, yeah.
Thanks. Essentially, the way the scores are compiled here, here's an example for the calculation. In fact, the calculation is normalized not according to sort of maximal scores, but on the tree. Okay? Because, again, different branches have different lengths. And for different branch lengths, the random background, thank you, the random background probability is also different. Okay? Now, let's look at a particular example. Say that were the experimental annotation of one particular tree for one particular annotation. And let's say the green ones were the ones that we predict. How do we measure how well we do? So the simplest, and again, the level of detail grows in that direction. So we could argue all that we want to do is really get the most detailed annotation out. So essentially we could even do a binary, we get it or we don't. Right? In a case such as this one here, where we have both that lead to this final one, we don't predict the final one, both that lead to it, we may argue this is still better than 0. But you're beginning to see that it's not that easy. There is no one solution in which we can completely convincingly argue that this is the right way of evaluating it. Okay? We can compare these two, the red, the observation, the green, the prediction, in different ways. And you can argue for many of these ways why they would be right. Okay? One extreme way is essentially you look at a recall versus precision. So of all the terms that you could have found, how many did you find at a given cut cut in the tree versus precision at that cut. How many of the ones that you actually predict are right? So counting the green ones here, this is wrong, this is wrong, this is wrong, this is right, this is right, right? And recall would be if I cut my tree here, I would miss those 3, right? Since we have 4 in this particular case, I would have 1 in 4 in recall. Okay, now we look at 3 different outcomes, or 3 different methods, and we could have 3 different curves. Now one way of comparing them is by saying, well, we take the one with the highest area under the curve. Thanks to and quiet from here on, but now you will be the bitten one. What else could we do? So again, one way is we could say the highest area under the curve in this particular case most likely is P1, so P1 is the best. What else could we do? To distinguish, or to say method P is better, or method P2 is better, or method P3 is better. Well, we could fix a point. We could argue that we don't really care that we get all the terms out of that are experimentally observed, but we do care that the ones we predict have a high reliability. So we're looking for a realm where we have very low recall, possibly, but high precision. So let's just go for the method that has the highest precision. In this particular case, it's all easy because it's still P1. We could also go for some F value, right? That is essentially the harmonic mean between recall and precision. And this, in fact, may be highest somewhere here, and we could look for the max in the F. And we can essentially look for the average. Why would you go for the average rather than the area under the curve? Much easier to compute. As simple as that. The max I talked about already. So in this particular case here is the average over the 3. So the max F score, that's the harmonic mean here. I talked about that. And now, essentially, how do we evaluate the method? We need some random subset of the ones for which we know the annotations, so some random subset of today's database. And then we sort of try to see how well, for some subset of these, we can in cross-validation do that. Now, let's get back to the 3 methods, and I should have put this slide in. So I defined a problem that I felt was very well defined. I defined it in a way in which I clearly would write the method section in a paper. And we had 3 groups that did different things. And they all did, in principle, implement exactly like you immediately translated what I said to the number 5. You could take 5 nearest neighbor. I never said that, right? But somehow it is so easy we all map what we see to our internal understanding, and the fact that I had 5 made you map it to that, and it is reasonable. So the next problem was that the different solutions, I flashed them to you. When they flashed them to me, I was first surprised because it was not quite. I only wanted the first. That would have been the easiest thing. And then finally I realized why I took them until Christmas, because they had gone through much more complication than that. But then I looked at them and I could not tell whether they're different. I thought it wouldn't matter. Okay, here are the results. Those are 3 different methods: green, purple, and blue. The others, the random is this background here, and it's a recall precision. Now, here we get to another issue. So that was done here on whatever the method was assessed. The students assessed their tools on different methods, different targets, simply because for some it didn't work, or for some their method didn't work. Or here, this is the subset where they all have the same target. Okay, that's a much smaller subset than that. They're not that different. When you look at random, you see they're relatively similar. But what you clearly see, and meta is not really important at the moment, is a path through them, but what you clearly see is that green, cyan, and purple differ. They occupy very different areas in the rock curve, right? And this is not clear to me. So first of all, I did not see that they would differ. And secondly, I did not see that one of them would be best. So now, by some argument, you can say the green one here has only 2 points. So it does not really have a differentiated score. This is why it doesn't scan the rock curve. But at least those 2 numbers that it has are above anything comparable for the other 2. So it clearly picks up the signal. And I could not really tell from the method description that this is the case. We were surprised how well the nearest neighbor worked, that you immediately translated. And we were surprised how much the technical details mattered. And then, the next step was, and that is already on here, we tried to somehow find an optimization through those 3. How could you do that?
Combine them.
How?
Well, you see that there are different areas on the recall axis that they are best the individual methods. So you could. No, that doesn't make sense. But
unfortunately, wait a moment. I want to hold on that, because this is. Why do you say it doesn't make sense? So let me explain to you why I want to push on this. This is a way in which many people tried and believed it would work. Did you really figure out why it didn't work? Many people have tried exactly for years what you're saying. Did you really immediately in 1 second stumble over where the mistake is? Why doesn't it work?
I think that the recall is something you assess also with the method.
You don't see it.
Yeah,
that's the problem. The method does not tell you where you are. Right? So from the output of green, you cannot tell, for this one I take green, and for this one I take purple, because this is an after I know the answer assessment. All right? So that's a dream. But many people have followed this dream because, well, we don't read it off the method, but maybe there's something in the method that would tell me whether I'm here or there or there. In particular, I mean, the ideal point is this purple and this blue here, right? There are 2 different parts. So combining those would clearly help. So again, even in my group, we have many people who have sort of tried to do something like that. Most of the time it doesn't work. Till.
Oh, yeah.
Okay, so what else could we do?
Combine the 3 methods?
Yes, that's what he said.
No, I mean, how we could combine them.
Yes.
I mean, what are the outputs again of the methods? So probability-wise?
Yes.
So one could average the 3 outputs of 3 methods of the probability output.
Yes, that could work, but if
so it's a kind of majority vote.
So, but look at these. It's almost disjunct, right? Pure average may not be the best way of doing it. In fact, I should have shown the pure average here. I didn't. So the brown is not the pure average. The pure average kind of situation, if you say you had the brown and say this would be 1, those 2 are easy to average. But something that's almost disjunct, like the blue and the purple and the green here, it's very difficult to average over. I mean, it's easy to average over, but it's very difficult to gain from it. It's also true that so the first solution that we had here, the brown one, does not gain over the green. It does not reach the maximal points. It only finds a path that is somehow a little bit of the best of all. That's what it is. But how could you do it? What's the simplest thing that it can do other than just averaging numbers?
Max? Or like use the max
yes,
of all functions?
Actually, that also, so okay, I'm sorry, I have to step back. Because I mislead you by saying the simplest thing. It is not simple. So the simple things all didn't work. The simplest things is what we tried. So this method meta here already used some machine learning. But then it's simple in the sense that all it uses is the input of the other methods. And that is still essentially very simple. So we're not bringing in any features, we're not bringing in any novelty, we're not machine learning in any complication. Essentially, you have 3 different input units, and we try to find a path through a probability landscape mapping. It's relatively simple in that sense, right? But it's still a machine learning device. I believe it was a linear regression that was done for that. Okay, let's get back to the CAFA idea. So the CAFA idea was, okay, we know in biology that everybody uses methods to predict function, but how we typically do these kind of homology-based predictions, we do all kinds of other methods, but how well do they do? And the idea was just, collect data from the last year or so and run prediction methods for those data that essentially have not been seen to the methods. Use different data sets, different target classes, different measures, and see how far we get. That was done in 2011, and the people who started it, there was Predrag, Ido mainly. By the way, this is the man behind SwissProt, so the database of all experimental annotations, and Sean. So those 3 here were the original people who drove CAFA and essentially tried to have a competition that assesses how well methods do. And the rest here is just names for the board. So find organizers, done. Now we need to agree on target functions, and essentially we already talked about that. We take Go, the gene ontology, simply because that is sort of the most comprehensive set of annotations with numbers that we have. There are a couple of problems with target functions or with Go ontology. One, for instance, all of this is sort of essentially, it implies something different. But in terms of a Go number, it's just a number. How do we differentiate between those? Positively, negatively regulate is much more precise than is part of. Okay? So how do we give different weights for that? But the moment you start with weights, you put in your own identity or your own idea what you call better, right? You cannot do that. Okay, then there are different ways in which we get relations between parent and child inference rules. So this is one way, and this is another way. Part of the example that I already showed you. It should matter somehow which of these paths you follow. There are different, what is called evidence scores. And one particular evidence score that is highlighted with this arrow here, all of these are different evidence scores that are used by Go. Evidence scores means there's this Go number, I say is an ADH1, and I say that because of evidence this. Evidence this could be inferred by experiment. That sort of appears to be relatively clear, depends on the experiment that people do. But you already see that it gets more complicated. IDA is inferred from a direct assay. Well, a direct assay is a direct experiment, so what's the difference between 1 and 2? And you already begin to see that in the detail things are really complicated. And one particular one here is inferred from sequence orthology or similarity. This is exactly what we're trying to measure here. If that is used, the kind of rationale that we're trying to see how well it does, if that is used to in fact infer, then we're turning full circle here, right? So that's another problem in this story. Then there are different qualifiers. Contributes to, color-localizes are not contributes to. They are, again, so not being that Go number. How do you classify that? So you have a particular, you say this is not an ADH1. How do you use that information? It is important. So you make 1 point from all the typical enzymes that you could have. This is not it. How do you value that? And then there are different Go versions. So here's just a couple of Go versions. They actually change within days sometimes. There are different databases that annotate Go versions. So SwissProt is the database of experimental annotations. Then there's the Go database that essentially is taken from SwissProt. There's a Go annotation database. And you see that essentially all of these resources are connected. There's a large overlap. But there's a lot of differences too. So what do we take? Do we take the union? Do we take everything? Those are issues that we have to solve. Yes?
How's this work? if SwissProt is the database for all annotated proteins, how does the Go database have something that SwissProt doesn't have?
Now it doesn't. Okay, so before you actually look at these numbers, what you see in the paper is we take the annotations from SwissProt.
Yeah.
Okay, so it doesn't. Then you begin looking, and then, well, I mean, in some sense the numbers are relatively small, you may argue. So at least the ones where SwissProt is really unique, maybe SwissProt just added that over the last month.
Oh, okay.
This is one explanation. It's not always going to work that way. So this is, when you talk to the SwissProt people, that's what they immediately answer. That makes a lot of sense. But there are other cases too. They have been overlooked. It could be that the Go annotator felt like this annotation is not sufficiently validated or read. There's a variety of reasons. The ones that are original here could be that they come, again, they to some extent come from here, but here are still 1,200 original ones, and they often are those that have evidence core sequence homology. So the story is more complicated.
So would you say that the Go database is higher quality in annotation? No.
So the highest quality database undoubtedly that exists is SwissProt. And that is when I met, so again, you have different, when you say quality, again, you have sort of an image behind of what you want. One quality of a database is that every entry I look at, there's a number, there's a qualifier that says this is an ADH1. This should be right. I define that as quality, right? Another definition of quality is when I see ADH1, it gives me all the links to the right papers and says ADH1 if and only if there's lots of explicit information. Okay, this is another, sort of is a related measure of quality. The third sort of quality measure is, let's assume you have a database that has 50 annotations for 20,000 human proteins. Those 50 annotations are really good. But would you call that a high quality database? Those 50 are good and the 50 are right, but it's actually nonsense. It's not a high quality database. So by saying that, you already begin to realize you could also call coverage something like quality. So if you have too few, obviously you can never be a high quality database. So you need also a certain number. You see, you certainly need to somehow be up to date on the literature. It's not directly what you expect as quality, but it's something that you somehow expect. Okay, and then there's the third, that is my direct answer to you, which is sort of the comparison of these three circles, which is somehow the dynamics. So how much does the database add in a given time point? A good database should add a lot. Again, this is tens of people who sit day and night, not day and night, but essentially sit there all day long and annotate. And this is true here, it's less, a little bit less true here, it's more, so here's more computer, here's more really expert annotation. So you also expect that things change. Right? You know, these people work for a year, and at some point SwissProt had 100 people on every time point working for them over an entire year. So there should be a difference, right? So this is also in some sense quality. Anyway. Now, this led to some sort of simplification. Initially, the organizers focused on two different gene ontologies, namely the molecular function, so this is enzyme, and the biological process, this is the pathway, glycolysis. And they had essentially those days when in 2011, these were the numbers of leaves and terms they had in the then accepted Go tree. Okay, then we need to agree on targets. How can we do that?
Just take a lot of targets.
Take a
lot of targets. Just
yes.
Like a large amount. So
where do you get them from?
From sequences.
If you took them from SwissProt, anybody can cheat.
No, but you have a large database of sequences with like
yes.
60 million. You can just, I don't know, grab a bunch, like 10 million.
That is a great idea. So you can essentially take whatever anybody wants. The problem with that is some people have methods where they only have a certain throughput. So we have to sort of clock this somehow by time. So in the CAFA experiment that you're now part of, the time, I believe it started a few weeks ago. It runs until shortly after Christmas, roughly. And then it's the end, right? That's the method development time. And then there is a sort of target, or the accumulation of experimental result time. And then there has to be an assessment. And then there's the meeting and so on. Okay? That's essentially the way it goes. So now, we cannot run this necessarily on 60 million. Or we could have one competition, and we actually in CAFA 2 we did. They did.
I was just saying the numbers, so the probability that there would be some experimental evidence was very high, close to 1.
How do you do that? That's an interesting idea. Anybody any idea how we can do that? So we want to have targets that we can be almost certain that somebody's going to have publication on over the next N months. How can we do that? How could we possibly do that? Any idea?
I think in the field of this research are maybe some current proteins that everybody observes and thinks about. So the hot proteins are...
Yes, that is absolutely right what you say. And in fact, there are most likely something in the ballpark of 30, 40 hot proteins at the moment. But for these hot proteins, it's very difficult to see how much already some groups have knowledge of. So the hotter they are, somehow the more you want to stay away from it because of the complication that somebody could already have an answer and it could have gone through. Any other idea?
It might be too fancy, but what I'm always thinking is like a Gaussian mixture model would work. Like we have like 40,000 that we have annotated, but we know that there's a lot more proteins like that we have a string for. And so we could try to understand why we have results like
so what's so special about
experiment results, why we have it for them, and trying to separate that group. And
I should talk. I will bring in a talk about the dark proteome. That is a very interesting story. But I'm afraid this is going to be, so yes, I believe what you're saying ultimately is, when we understand what the bias in today's databases is, then it will tell me something about today's experimental methods, that will tell me something. Very, very true. Slightly different story. Here, in fact, the story is yet different. What I'm asking not is how can we predict. The Gaussian mixture is still a different story. Maybe we should do that for CAFA. But here the question really is, where to get my targets from? So where do I get those proteins from that will essentially be given to the methods that predict, and will then in the meeting in July, the CAFA 3 will be in Prague, will be assessed in Prague. How do I get the targets? So the one idea is we get the hottest. That has a problem because some of the hot ones, there are so many working on it that you never know how much is known. That you get 60 million, just take them all, essentially. The problem with that is the number for some methods may be too big. So we need something that is sort of in between these two extremes, or any other idea that somebody can come up with. So I had a great idea that actually didn't work. And
well, you could ask some researchers to do experiments for you.
No. So this let's get back to this experiments for you. I'll get back to that toward the end of the talk. When experiments for you. So what we actually did in a different context to predict protein structures, CASP, was to talk to experimentalists. What is in your pipeline? What are the structures for which you, over the next half year, believe you get a structure? And that's the way CASP runs. And CASP, on a cycle of CASP every 2 years, has about 100 targets. And that works. Here we wanted much more than that. So the idea that we initially had was to go aggressively to the journals and ask them to give us whatever is about to be published. And then we contact the authors. Unfortunately, that didn't scale. So it worked for some, so we had a trial phase in which we tried that out. And it was easy to reach 20, 30, 40 with that. But then things went wrong with the journals. And anyway, so this was not enough. So yeah?
The other way around, maybe. So you
finding people?
No, people find you.
Yeah.
So if I'm an experimenter, I'm a nice guy, and I just contact you that I'm about to do something.
That means that everybody knows about you. So you need so this, maybe we could actually try something like that now. So after the 3 CAFAs, or now it's the 3rd, maybe we could have actually tossed something like that in. That's an interesting idea. Maybe at some point we should also talk about the midnight zone or the moonlighting proteins. Not sure you guys should handle those, but I believe those are contributed by the community. Till?
Maybe one could have a different approach from like seeing how much request is for the protein in the database if you have like a website.
If you look at the database, then that is, yes, in fact that was one of the Predrag ideas. Okay. The problem with that is, at that moment where you say that, and in some sense it's like your hot proteins, you focus on exactly the proteins that are already there. What you want to estimate is how well would you do for something that nobody knows anything about today.
Yeah, I mean like where you take out the results where it actually would provide information, but if you hand over a string and the database does a checkup and doesn't find anything reasonable, then you would only like take from that subset.
So that was
requests,
in fact that was another way in which they did it, is they asked the database annotators. So to simply give them, I take that from what you say as an implicit part, and that did work. Another thing that worked is they took the model organism initially. So we went for a larger set of, I can't remember what it was. Clearly human was one of those. So it was not 60 million, but it was 60,000 in that ballpark, somewhere in between. Okay, so again, we already discussed that. So there's the prediction phase, there's the annotation phase. Annotation means I wait for annotations to come in. And then there's an evaluation phase, so I compare these two, essentially. Now, those are annotation of proteins existing. And so again, this was something that Predrag did, Radivojak, initially to see whether that would be reasonable, and that essentially, one way of doing it is, this is the number of things that in principle you could get in a month. So that looked like ample. We didn't get as many. So, bless you. Bless you again. This is the number of targets we got. It's a considerable number. So now most of those, unfortunately, are evidenced by inference through homology. So this is chase your own tail kind of story. MFO is molecular function, BPO is essentially the pathway, and the targets are here split to how they distribute, and how they distribute also in terms of number of details. And it turned out that most of the stuff that we had was in fact not that detailed. It was on a very high-end level. It's a kinase kind of thing. Then a lot, for instance, the protein binds. Okay, some proteins don't bind. But it's not exactly extremely informative to know that the protein binds. It binds DNA is a different story. Okay, but protein binds. Protein has some zinc ion bindings. Protein, in fact, zinc ion is a fairly frequent thing. It has some homodimerization activity. What it means is that you have some evidence that it forms a dimer. So that, in fact, in order to aggregate, it has to be a dimer. ADH1 I showed you in order to act, the enzyme has to come together as a homodimer. Well, we need to know that it's a homodimer, otherwise we cannot do our experiment. But knowing that it's a homodimer doesn't help us at all, right? At all is not right. But it's not very informative about what this enzyme actually does. So you see somehow that the dominant information is, in some sense, also the one that is least accurate or least detailed. The next issue here we have the distribution again for molecular function, for process here. How similar was the one that was newly determined, so that was added to the database for which I had a new annotation, how similar in sequence was it to something that had already been annotated before? Okay? And you see that essentially a lot is in the realm where we would argue this is exactly what we are trying to do here. This is what we call the simplest model. This is sort of covering some point here, in fact, almost the entire MFO. So most of the ones that had been added had been similar to something that was already there. How can that be? So the question that I'm asking now has nothing to do with logic. It's, in some sense, I would call it a social reason. I mentioned it before. Does anybody remember? Ultimately, how come, so we have one protein here in this particular case, we have an annotation in the database, we have an almost identical protein, and we publish a new result.
Because you're forced to publish as a researcher? Publish or perish?
I am not clear. I'm unsure how much of the reason that is. That certainly is most likely the most dominant one. Yes. So you do it again. You have to get by your referees. How can you get by the referees? Well, it's slightly different. You change one residue, it still binds, or something like that. It's one reason. That's, let's call it, in some sense, a negative reason. This is the way science works, and negative and positive are the wrong words for that. But there is, let's call it a good biological reason too. I believe this is dominant, Nicholas. But what else could we think about? I mentioned it. So again, doing the experiment in the prokaryote, in a bacterium, is easier than doing it in mouse, fly, or even human. Human is much more complicated because of all kinds of legal situations, because of the difficulty to keep cell cultures alive, the technical difficulties to keep cell cultures alive. It's much easier in a lab to grow E. coli bacteria and keep them happy than to grow human. You can take, I don't know whether you know that, so my wife is doing that sort of thing. You take a piece of the skin and you can grow a culture. A human culture, right? The problem is it's not going to be there for a year. It has a limited time span. You do this with E. coli, it's going to be there forever. It's nothing. It's very easy to maintain that. So for the human cell culture, my wife has essentially to go almost every day to the lab and just add some reagents or some stuff to it to make the cell culture look happy, feel happy. Anyway, so that's a reason you first do the structure maybe in mouse or maybe in a bacterium, and then you do it later in human. So the human publication is still a very, very important one. So this is not publish or perish. This is, everybody had been waiting for the human protein. But ultimately on the protein level it's exactly identical. It's very easy. It's still a good reason, I call it a good reason, because you had to make sure. Sometimes function changes. That's orthology and paralogy, function changes. So we have to make sure before we go into treatment, we have to make sure that really is the function that we target. Okay. Now the third one, however, is somehow in between these two extremes. So maybe somebody had done the function E. coli, and somebody else does it in a different bacterium that is related to E. coli. It's much easier to do, because I already know where to look. It is publishable, because it's a different bacterium. It is slightly different, it's not trivial. Nobody really cares except the people who work exactly on that bacterium, which may be a minor group. But still, it is something that will be added to the database, and it's relatively easy to do. That's another reason. So it goes into the publish and perish. It adds relevant information, not that much. But all of these are reasons why this actually happens. So now, the next issue really here is the evaluation measures. And I'm not going to be very good going through this, because ultimately, because I'm still not convinced that we have been doing the right thing here. Now we're in CAFA 3, and this problem we still have not solved. And then we evaluate the predictions. So that is the issue again. The average over the three methods, there's a max F1, that's one way of evaluation measure. In fact, that is the one that was used for CAFA 1, the max F1. The positive aspect of the max F1 is a method, so the three P1, P2, P3. Remember I told you that the methods spit out not only one prediction but three, for instance, right? And so for these three different, you have a top model, a second best, and a third best. And those are your three lines, and somehow you average over those three lines. If you only give one best method, and if you know that this is P1 is really your best method, then you win. But if you can't say that and you throw in many things, then it will be averaged over you, and this is fair, right? So the average over those three methods is something that is fair to the competition, fair to the developer, and picking the max score is extremely fair in some sense, right? for the method developer. You pick whatever is this method best at. Some methods may be best at getting high accuracy, high precision, lower recall. Some may be better at getting everything at a low precision, right? And when you develop as a method developer, develop that, and you believe this is the strength of your method, you define it. And in that sense, picking the max F1 was something that tried to be as fair as possible to the predictors. And then we have to evaluate the prediction methods. So those are somehow ways of classifying them. And you see already what we need to do in order to do this here is we do an alignment. We need to compare the unknown protein to the database by an alignment method. And every method that contributed somehow used that at some point. And then they used all kinds of other properties. Sequence properties, somehow do you have charges in there? Phylogeny, how does it look in the genomic context, in the family context? They looked at predicted protein structure. This is just the same in more detail here. What I'm missing here is genetic interaction and protein interactions. So you could also use protein A interacts with protein B, protein C in order to refine your prediction of the function. Because say you believe it is most similar to number 5, and now say you would know that for the experimental case for which you know this goal number 5 is for a protein, say in E. coli, and for that protein in E. coli you know it has 3 interaction partners. Say U is a human protein, and you believe that this E. coli protein has the similar function. Does your human protein have an interaction with the sort of orthologous or the similar proteins in human? If so, then your trust that it is in fact Go5 increases tremendously, right? And that's the rationale behind here. But in a more statistical sense, so, well, not in a more statistical sense. So does it have the same interaction partners as one of them? Another one is, of all the possible interaction partners, what's the overlap in the statistical sense? There's a certain probability, and that increases or lowers your score. Okay, then there was another step made here that only one model was evaluated, so no averaging. I couldn't remember that. Then they were separately done for molecular function and process. That's trivial. You cannot join those. And then we also looked at priors. So prior. So one prior is, so I predict that a particular protein is a kinase. So of all the proteins in human 20,000, how many are kinases? That's one prior, right? So what's the random hit probability of BLAST here? Essentially is something like I described in my lecture, much simpler. Just run the simplest alignment method, pick the top end. And that is another one of the background, right? Because anybody can do that. So you have to, any method that you develop has to be much better than that. It has to be trivially better than a prior, but it has to also clearly be better than the trivialist thing that anybody can develop. And here we are. This is one slide of the results. And as you can see, this is the problem with these methods, or these kind of comparisons, is they get immediately very obvious. So one thing is obvious, you see differences, right? You see also something that is highest here, not quite as simple. You see some things that have very high precision. They are above this green one here. You see clearly things that are very low. So you somehow looking at this, you may feel that you see some winners, right? And these winners, sort of molecular function, biological process, that's an average here over different methods. And somewhere for the process down the line, that is the average over the 3 groups after machine learning, the so-called meta student. That's where it landed, somewhere in the middle of the field. And here, I can't remember where we are. I guess we are on that one here, so below the middle of the field. Now, essentially there are top 3 methods here, and the way they were best is by using more data. The top method uses the interaction, uses text mining, so are there keywords that are related already there. It uses sequence, gene expression data. The other 2 here, essentially, or at least the second best, if you compare the second best and the third best, so the third best really did something on top. So text my, other than that, they did exactly the same. But this did not quite help them. In fact, they got a little bit worse. But that may be because they didn't use the alignment idea well enough. So I don't really see where, so here's, by the way, the naive and the blast. Naive is just the background. So here's another thing that I'd like to point out here. So this is naive. Naive essentially is, what's the probability of getting a kinase by simply counting how many kinases do you have in your database, right? Simple statistics. That's the blast. Doing this, finding the top hit. How can they be similar?
They are similar. Blast is worse.
Fair enough. But within the, so when you look at the error estimates here, they are within each other. They're within less than a standard deviation of each other. So I'd argue in a standard error sense they are distinguishable. But you're right. So it's even, if anything, then BLAST is worse. So BLAST uses some information. BLAST uses an explicit lookup. This is the dumbest thing you can do. Probability of kinase according to how many kinases I have in my database. Has nothing to do with my protein that I have. It's the same for any protein that I will see. It's just completely random. This is specific. It may not work very well, but it's specific to that. Find the one that is most similar, and the one that is most similar, in most cases you saw in the distribution, most of them were somehow similar. There was one in there that was most similar. There was one that I could have found. In some cases I may not find it. But how can they be so similar to each other? And ultimately the answer to that question is the way, so let me alert you to this kind of thinking. It's not about CAFA, the point that I'm trying to raise here. This point is about when you see things that absolutely do not make sense. Well, the first thing is you hunt your bug, right? It's like outliers. Is there anything that you made a mistake in programming? Very often the answer, you will find a bug this way. But something else is here. If something so unreasonable happens, it tells you that your evaluation measure is not quite right. There's something that went wrong. So picking the max, picking wherever you have some overlap, going in the Go tree to the highest level, essentially the kinases, you essentially, it turns out that what dominates here is exactly the highest level. So you're essentially not doing much more with the blast. And then you pick the max value, and somehow you do too much of an optimization. What initially, when you look at, I described the evaluation procedure. I didn't describe it in detail, but if I had enough time, I'm sure I can convince everybody in the room here that this is very reasonable. All the CAFA organizers were convinced that it's very reasonable. It made a lot of sense. This is the moment, after 2 years, when it dawns on you that something went wrong. Your measure cannot be right if a random dart throwing is as good as something that contains information, right? Now we can also, unfortunately, so in CAFA 2, what Predrag did is he tried to sort of find measures that are more informative in that sort of thing. That leads to another level of optimization that is not necessarily the right path. And we still, as I said, we still haven't found the right way. Another popular method is GoStruct here that did not compete because it was in fact the method from the organizer and therefore didn't compete. But anyway, so the integration is the answer that worked. But it seemed that it works somehow well, but there was a lot of room for improvement. So here's one way in which we improved that. After the CAFA, we realized that actually there was a bug. There were other issues that we had, and we re-optimized. So, but now I'm going to show you, from here on, I show you results that are fake. In the sense, I'm going to show you something that is like CAFA, was derived after we knew the answer. This is the way you always develop prediction methods. So in that sense, it's clean, but the idea of CAFA is to sort of go out from the trouble that we all too often sort of glance at what we try to be hidden. So after CAFA, the number of proteins had increased, And the idea simply was take new proteins. New proteins have better annotations because new experiments are much more accurate than older ones. And let's redo the CAFA. And in fact, let's redo the prediction method that we had, the optimization of the three original methods. And we did this by sort of having three different phases to, in fact, split today's database into different time frames, allowed us to have some for training and some from different time points, originally in time, when they were added to the database, gave us a different slice that we could use for testing. No more cross-validation. We hoped that through this, essentially we would like simulate a CAFA experiment. We would simulate an experiment where what we have for training is this, and what we use in order to do optimization of free parameters is this, and what we use to assess is this. Now let's go into, I will come back to the way of calling it. So there's training. I like to refer it as cross-training and testing. What I mean by this is, think about a neural network. You train a neural network, the neural network has a certain number of hidden units. Then you think about an SVM and you think about a linear regression. And you get three, the NN gives you, say, whatever, 70%. The SVM gives you 72%, and the linear regression gives you 74%. And then typically what you do in your thesis is you say the accuracy that I reach is 74%, and the best method is a linear regression. Now in doing that, and typically the way you do that is you do a full cross-validation. All of these numbers are done by full cross-validation. For cross-validation, again, you have a data set, you split it into, say, two parts. Use that for testing and that for training. You do a random split and you rotate it around. We will talk about that again and again later. But in principle, this is the way you derive these numbers. So what's wrong in it? What's wrong with this final argument? 74 is the performance of my method, and the best method is linear regression. What's wrong? And again, we'll talk about that again. Do you see what's wrong with it?
I mean, the result is based on some data.
Yeah.
So if the data changes, maybe linear regression won't be the best anymore.
That is, we get into that. No, in principle, you believe. The idea of cross-validation is you massage your data in a way that is optimal, such that you believe you can give an answer for tomorrow.
Yes.
You're right. You may be wrong. But in principle, that's not what I mean. Because that is the problem for all three of them.
I just think about if they're really data changes, maybe SVM is sounding better. But
that's a very emotional answer. But you can also give, but still 74 is higher. Here's another thing that I believe is important. When you give me your answer, it's easy to give you your answer. If you put €100 on the table and we bet on your answer, you get more careful typically, or whatever the number is that you need to put on the table to be very careful. But I bet you, the higher the number is, the more you will end up with the 74 number, right? Whatever you say now. If you were to put €1,000 on the table and you would bet which of the three is the best, you would always go for 74. Or maybe you would go and say, well, you're pushing me in a way, I don't want to give my €1,000 for anything. So the number of euros that I'm talking about, it must be something that really makes you emotionally involved, scares you. Anyway, so you're too intellectual about this at the moment. But what I mean is something else.
Then the accuracy itself.
All of that we will get back to.
Best measurement. I mean,
we will get back to that, and that's also a point. But there's something more wrong, and that goes back to this, which is, in principle, cross-validation means you come up with an answer that is valid for the future. Means that you are giving an answer for something that you never have seen before. Meaning you pretend that this answer is valid for the part that you have been hiding. Right? But now you actually have it in the open. And you say this, not that. And you do that in the open. You suddenly use your data. You suddenly use what you were supposed to have under the table. You put it up and you say, now I can choose. But in choosing, you make use of the data. You're not supposed to do that, right? That's exactly what you need the cross-training for. So the two-way split is not enough. You need a three-way split, okay? I call it cross-training. Again, there are so many different words out there. I feel it's easy to call it cross-training because it immediately reminds you that this still has to do with the optimization process, right? So typically, the training then could be you do the parameter optimization for these three different methods, and on cross-training you say which of the three is the right answer. Or in the language of a neural network or an SVM, you train the SVM and then you look at different kernel functions for the SVM. Or for the neural network, you look at different levels of hidden units or different architectures. And you say which one is best. And all of that you do at the cross-training. And only then do you use the test set that you had never seen before. And then you publish these numbers. And if in your publication, say, and this is something, bear with me, you will see again. So in your publication, say the actual test would be this. And in the cross-training, you would have something like 70, 76, 74. You would still have to say this is best. Right? That's the point. So in the cross-training you see that in fact the SVM gives the best result. But you will not be able to say, so the clean way of doing it is to say, well, I saw the best result for the validation set. No, wait a minute, it's the other way around. If that's the I'm sorry, just the other way around. So you would have to say the best is the SVM because it has, so the way I choose my model is according to the cross-training step. Cross-training is high as 76. So I say this is best and I publish this is my best method. But it turns out for the final test set is less, but still this is my best estimate. And again, in the €1,000 idea, in principle, if you think that through, if you bet for the CAFA prediction of tomorrow, you should actually set on the SVM. You don't know what went wrong with the test set. Maybe this induces in you to do additional steps. But the best thing, if your process worked, is this. Okay? We get back into that. Yeah?
I still feel, I mean, what I took from it now is that basically first you do hyperparameter optimization by cross-validation. That's how you make sure that all the methods are able to perform to their best based on the hyperparameter optimization. And then you pick, on a more informed choice, you pick the best method. But it's still biased to the fact which parameters you would optimize for each of the methods, I feel like.
Implicitly you say a couple of things now. So one thing that you say is essentially no matter how I do my cross-validation, I will not get out of, if I got trapped because I made a mistake in the design of my method, I will not get out of that. So mistake could be I sort of incorrectly optimize some parameter, right? And this problem will, no matter what I do my cross-validation, I will not get out of that. True. The only thing that we're addressing here is not to get the best model. What we are optimizing here is to get the best, the most optimal estimate for the model that we have. And that turns out to be the major part of the problem. Machine learning typically hangs here, not in doing the model, usually. Most people who do machine learning spend most of the time in getting the better model, but that may not be the right way of doing it. So anyway, we did a post-CAFA optimization, we did a machine learning optimization, linear regression optimization, and the method, things are getting complicated, but let me give you my word for it, the final method ultimately did better. And so these are some of the some of the positive things and some of the problems persisted. And we got into CAFA 2. I'm going to quickly gloss over it. So when you do something that sort of changes the field, then many things change. So for instance, the Go annotators began to change things. So databases changed according to the way predictors predicted, which is not quite what we wanted. But clearly it helps for them to look better, let's call it that way. Now, there are many things that happened. The rules changed. We looked for different proteins. We slightly changed the criteria. We had a much larger set, different types of proteins. We had a more mix from other categories. We also looked at HPO as human phenotype ontology that ultimately relates to diseases. That was tried to predict here. Still a small set because this is human only. This is all bacteria and everything else. There are different categories, different annotations, dataset statistics. We still have many that are here, but now you see that there are also many proteins here for which we find new annotations that are more distant, that are not so evidently clear. It's not so great on this one here, but still. Now here's another minor point. Now in the CAFA 2 121 methods applied, and most likely, so it went up from 34 to 121. Most likely in CAFA 3, which is what you're contributing to, it would be much more than that. And again, so looking at recall precision curve, that's naive here. Now we are getting at a measure where at least this problem is out. So naive really is clearly worse than the blue, the blast. There are two different blasts here, and there are some issues why they differ. But the sort of regular prediction methods are all up there, and they're all doing much better than the blast. That's for molecular function, so it's an enzyme. This is for molecular function for different classification. And so this is again the same method that we did before. It's the same thing that we did before, except for we retrained. Okay, and that's what we're going to do, I believe, this time as well. So nothing changes essentially, other than sort of fitting to a new set of experimental annotations. And overall, these are two different scales by which you measure how well you rank. Those are Swissprot EBI, so different annotation systems or groups that annotated what is right. 10 plus 14, the total number of annotations 28, and you see that's rank 2, rank 8, rank 4. That is the localization, and that is what I will talk about next week. Begin talking about next week. That's the process, so glycolysis. And you see that somehow the glycolysis, so the process is worse by this type of method than the function. That is an enzyme. Something like that is understandable. So which process you belong to, you want additional information which we don't use, such as interaction information or whatever else you can do. And localization, this is pretty high, relatively high, because it's a different method. It's not really this one. Overall, the meta student didn't perform too poorly. You do see molecular function, biological process, cellular function, human phenotype. The field is in fact relatively close. So again, this is an excerpt here. We have 121 of these bars. This is only the top and the lowest, so that's naive in red and the blast in blue. And here we have another example where the blue is lower than the red. So for the component, and the same for the human phenotype, where again those are new elements in the CAFA 2 competition, and we would immediately see that we again failed, right? While here, at least for the molecular function, now the problem of the wrong scales of this random background should really not bring anything for molecular function to work. Here also something still went wrong. And I believe I call it a day here. We saw, in fact, different results because we had different data bias. One of the major problems in biology really is the issue with bias. and in some sense the degree to which our data set is representing the universe of tomorrow is an important question. I made a mistake, so we begin on Thursday with the localization, and Thursday will be a chalk talk. Next week, Tuesday, is no lecture because of the student representative meeting. This is not the right one here. We'll get into prediction of subset localization next Thursday.
But Studentenvollversammlung, wasn't it last week?
No, it was Fashion.
Oh, sorry.
Yes. Thank you.